My name is Kira, and I'm an undergraduate at Indiana University, working as part of an international team developing computer simulations of how COVID-19 infection progresses in the body. Infection with SARS-CoV-2 presents many urgent scientific unknowns. How do the number of viruses and where they land affect the severity of the infection so much? Does the location of the initial infection matter? Why does the patient improve, only to then worsen with an overwhelming, damaging immune response? Why do men, the elderly, diabetics, and otherwise immunocompromised groups seem to have more severe patterns of disease? Since we can't do controlled experiments on patients, validated computer simulations of individuals can play an important role in accelerating therapy discovery and in personalizing medical treatment. Using models like ours, can we predict the severity of the disease in individuals? Can we use our predictions to optimize treatments for each person? Can we better allocate the limited medical supplies with these predictions in mind? Many of these questions can be addressed with virtual tissues. Virtual tissues are predictive computer simulations of the complex multi-scale interactions that occur within the body during the course of infection. For instance, primary viral replication, local and systemic immune response, viral clearance and recovery. The initial model isn't a representation of the specifics of SARS-CoV-2 infection per se, but it does make some interesting, potentially useful predictions. So far, our results show the immediate extinction of the virus by the immune response, as well as controlled infection after limited spread of the virus, and uncontrolled spread of the infection, leading to widespread tissue damage. Because of the complexity of the disease and the urgency of developing validated models, we are taking a collaborative, crowdsourced approach to building virtual tissues of COVID-19. To ensure the transparency and validity of our work, we are developing our models simultaneously with teams in Australia and in Germany. For ease of collaboration, it was crucial for us to design our framework to be modular. We started by building a computer simulation of a patch of epithelial lung tissue and the immune cells around it. We defined models of viral entry, replication, and spread. We also have implemented viral-induced tissue damage and cytokine signaling within the tissue. We then had a model of immune cell killing of infected and uninfected cells in the tissue, and finally, of the immune system and its response to and interactions with the infected tissue. Currently, we are in the process of adding models of the spread of infection in between tissues and of organ damage. Each module is designed to be interchangeable so that different teams can develop improved versions, swap out components, and explore consequences of different hypotheses. In the interest of crowdsourcing, our entire model is open source. Anyone can see what it contains, run it, reuse it, or modify it at will. This lends a dynamic approach to developing data at a rapid pace, needed in the urgency of the current pandemic. Our Model 0 shows 4 basic patterns of behavior depending on the following factors: the degree of initial infection, the rate of viral replication and level of immune response. As we continue to develop it, we hope to turn it into a useful and meaningful predictive tool. We want our model to help doctors triage what will be the difficult cases of COVID-19. We also want to enable them to design personal, individual treatments for their patients. Our team would very much appreciate your ideas and your participation in this project. You can find our latest models and download them to run at your computer at home, following the links below.